Okay, so I'm very happy that this is the last our colloquium speaker of APCTP colloquium program of 2024, and I'm very happy to be invite Professor Jülicher. So before the talk, I'm very quickly introduce his academic background as usual. Yeah. So, okay, so he studied the physics from Germany. So he started from the University of Stuttgart, and then he studied also in the University, Technical University Hochschule Aachen, and also he studied University of Cologne. And then for his PhD, he studied so he got the PhD um, so in Jülicher in Germany. So at the time, his doctoral topic was the morphology of vesicles, and then his supervisor was Professor Lipovsky. And then after he got the PhD in 1994, then he had some experience in some time, research time in Santa Barbara. And then he moved to Simon Fraser University in Vancouver as a postdoctoral researcher. And then he also spent some time in Paris, so Institute of Curie. So he spent some time, and he also, I think, he got some position in CNRS there, so researcher. Then he back to 2002 to the current Max Planck Institute in Dresden. So Max Planck Institute for the Physics of Complex System as a director. Since then, he's now the director of this Max Planck Institute, and also he's also the professor of biophysics division in the Technical University of Dresden in Germany. So as you know that Frank Jülicher is very well known and contributing a lot, many topics in biophysics. And I just point out that one thing. So 2023, he got the first the medal, IUPAP medal for the physics of life. And then this activity was held in Seoul in 2023. And also that's I'm very happy to announce this. So so that's I'm very brief introduction to Professor Frank Jülicher. So we welcome him, and then I also encourage that people anytime just ask a question to him. So then we can just more intense or very friendly atmosphere for during this colloquium. So thank you again for your accept of our invitation to the colloquium. Okay, thank you, Professor Jülicher. Yeah, you can start. Yeah.
Thank you very much for this kind introduction. I will share my screen.
Yeah.
Start the presentation. So can you see my screen?
Oh, yes. Yeah, very nicely. Yes.
Good.
Yeah.
So in my talk today, I will be talking about the subject of the physics of cells. And let me first briefly talk about the activities of my research interest, my research group. So we are studying the physics of living matter with an interest in particular on cells and tissues. And we're looking at biological systems as a physical system. And we're interested in particular in the spatial organization of cells and how complex spatial temporal dynamics emerges in living systems, starting from molecules, going to cells, and going to tissues. And since these systems are far from equilibrium, we develop a theory of active matter and active systems. In Dresden, we have a partnership with another Max Planck Institute. So we have our institute is the Max Planck Institute for the Physics of Complex Systems, and we have for many years a joint research program with the Max Planck Institutes of Molecular Cell Biology and Genetics. And in this collaboration, this joint research program, we have recently created a Center for Systems Biology that somehow aims to study complex spatiotemporal dynamics as systems behavior in biology. And this has now its own building, and several research groups sit there, and it also serves as a hub for us to collaborate with experimentalists. And more recently, in addition to physics, we also brought in mathematics and computer science to have a really interdisciplinary environment to study biology with many different approaches and concepts. And this is also done in collaboration with the Technical University Dresden, which is now also having more and more activities in these areas. I'd also like to start out with acknowledgements. So we've, as I mentioned, we have collaborations between these two Max Planck institutes. On the experimental side, we've been working for many years with Toni Heimann and with Stefan Grill, and I will talk about some of this work today. I'd like to highlight Christophe Sechner and Anne Grappin-Bouton. We have been working for many years on the theorists, in particular in Paris, with Jacques Pro and with Jean-François Journy. I would also like to highlight Christoph Weber, who is now a colleague in Augsburg, with whom we've collaborated a lot. And of course, there were many students and postdocs involved in the work I'm presenting, and cannot go through them all, but this is sort of essential for this work. So this is an outline of my talk today. So I will motivate what I'm describing very much by biological systems, and I will focus on one particular model system throughout my talk and highlight many different aspects. This model system is a very simple biological system that is very important for this type of questions, and it is very easy to work with experimentally. That's why it is in sort of at the center of this talk. And in my introduction, I will introduce some aspects. In particular, I want to discuss symmetry breaking of cells leading to symmetry breaking of organisms. Now, with this framework of cell division and cell symmetry breaking, I will then come to the physics of mechanochemical patterning as sort of the paradigm of how structures can form in a cell. This also will bring me to other aspects, such as emulsion physics in cells that organize biochemistry in a spatial manner. I will follow up on one aspect of symmetry breaking, which is chirality and left-right symmetry breaking. And in the last part of the talk, I will discuss sort of physical systems as minimal models for lifelike processes motivated by droplet physics under non-equilibrium chemical conditions. So let me go to my introduction, and let's start with some basic themes. So here you see a cell undergoing division. Here, some molecules are labeled with a fluorescent marker, and that's what you see highlighted in white. And you see here the mitotic spindle that forms, orients, and then is positioned, and it will then physically segregate the chromosomes in the cell. And after segregation, the cell can divide in two, and there are now two daughter cells coming from one cell. And some of this reveals somehow the extraordinary dynamics of a cell, and also the complex structures and patterns that form in a cell. Now, one can look at the same cell with a different technique without fluorescence labeling, just sort of non-labeled microscopy. And then you see different features of the same system. So you can see it's a fluid-like material. This fluid can set itself in motion. There is force generation, which leads to shape changes at the surface. There are flows generated, so there's some hydrodynamics going on. Now, one of the reasons why the system, while it divides at such a strong visible movements, has to do with the fact that the symmetry is broken, that the cell divides unequally in two different daughter cells. So the cell that we're talking about, and that's a biological model system that I've alluded to at the beginning, is a cell stemming from this little worm, this called C. elegans. And it's this worm lays eggs, and the very first cell division of this fertilized egg is this asymmetric division that we've looked at. The one that's interesting is the hermaphrodite, which can fertilize itself. So it's a biologically very elegant model system to study this very first cell division. This very first cell division breaks the symmetry because there are two different cells that form, and it already lays out the future head and the future tail. So it breaks already the symmetry between head and tail of the animal that will form from this cell. So this relates to the question of how does a fertilized egg sort of develop by dividing and then changing cell types to form an organism. And initially, this requires the breaking of symmetries. The egg is highly symmetric. The animal has broken anterior, posterior, front, back symmetry. It then also has a broken back, front symmetry. Now, in the external view, such an animal is typically left-right mirror symmetric, but the internal organs also break the left-right symmetry. So the organism itself has a broken left-right symmetry. All the symmetries are broken early during development in this worm. In fact, let me just briefly explain that for this model system because it will become relevant later. In this model system, all the cells have names. And so the names are, the initial cell is called P0. That's just a name biologists give to this cell. That's the fertilized egg. And it divides in two different cells by an asymmetric division in cells, one cell called P1, one cell called AB. That's the two-cell stage, which already breaks the symmetry between the head and the tail of the animal that forms. The two-cell state still has two more mirror planes. And the next, each of these cells divide again, and then we have four cells. One of the cells divides symmetrically in two cells that are very similar. They have, again, their names, which I list here. And the second cell divides again asymmetrically into different cells, which are also named here. And this asymmetric division now breaks the symmetry between front and back, dorsoventral. So now there is one mirror plane left. System now, if you look at a top view, we see the two AB cells that are very similar. We have this P2 cell here in the front, and the EMS cell is hidden below. It's not shown here, but the system is still mirror symmetric. And now in the next division, when these AB cells divide again, they undergo a symmetric division, which could keep the system mirror symmetric, but in fact what happens is there is a rotation of this axis, which now breaks the mirror symmetry and breaks the left-right symmetry. And this is always broken in the same way. So this system is always knows where left and right is. And this broken left-right symmetry is now continuing, and the final adult organism has a broken left-right symmetry of the internal organs. It's the same idea like in humans, the body plan has the heart on the left side of the body. This is also due to such left-right symmetry breaking during development. Now, this has to do with chirality. So one needs a chiral process to break left-right symmetry in a particular way. The chirality implies that an object is different from its mirror image. And if it's different from its mirror image, it's chiral. And this is, of course, something that is very common in biology. For example, in the example of a snail shell, it comes with a certain chirality, and organisms have a chiral body plan. And now this originates from the chirality of molecules, which is well known. Biomolecules are complex enough that they are chiral, and this has to be translated to the macroscopic scale to generate organism chirality. Now, in order to break these symmetries, we also need broken symmetries on the cell scale. So in order to divide a cell asymmetrically, the cell needs to acquire polarity, which means it needs to have a broken symmetry on the cell scale. And as I also show later, cells can also have chirality. So on the cell scale, there can be broken chiral symmetries. While cell polarity is typically reflected in a symmetry of chemical patterns, like sketched here with this red and green surface patch of a cell, which defines a vectorial asymmetry. Cell chirality typically is revealed by flows that are chirally asymmetric, as I'll show later. Now, let's come a bit to asymmetric cell division and how these asymmetries are established. One can reveal cell polarity by labeling certain proteins with fluorescent labels. And you see an example here on top. There are two proteins labeled different colors, and you can see that the cell is polar. It has a different protein on this anterior left half of the cell surface as on the right side. Now, as the cell is polar, it can divide asymmetrically, but this asymmetric division, interestingly, it leads not only to cells of different size, it also leads to different membrane composition after cells divide, because these membrane surfaces are different. But also the cell interior, the cell cytoplasm, is divided unequally. This happens via first gradients of molecules that form in this polarized cell. So there is concentrations which are higher on the left or on the right. And also there are structures, they're called granules, but they're in fact, as I'll show, droplets that are segregated to one side of the cell. And as the cell divides, only one daughter cell receives this material and not the other daughter cell. So the asymmetric cell division involves a segregation of material, both in the membrane and in the cell interior, as the cell divides. Now, how is this cell polarity established? What happens is that initially the cell is not polar, and then there is a trigger, which typically comes from the entry of the sperm, that triggers a symmetry breaking process, which drives first flows on the cell surface, that then leads to the buildup of the second domain in the presence of the initial one. So there are surface flows, and of course these surface flows then, by coupling to the bulk, also generate bulk flows. And so there's a hydrodynamic flow profile during this pattern, during the symmetry breaking process. So surface flows and cytoplasmic flows, and all this is driven by active processes that happen near the cell surface. So I show you here a movie of this process. So this is a fluorescently labeled cell with two different labels, and here you see essentially there's one color on the surface, hard to see right now. The second color is inside, so we start from a symmetric case where the outside is all the same color. There's some trigger happening here, and now suddenly there are these flows, and we see one domain forming on the left and the other domain forming on the right. And now the cell is polarized, and now it can divide asymmetrically. And this happens on the surface by a contractile material. So there's a surface material near the cell membrane that can contract, and by its contraction, it generates these flows. So this is an example of a mechanochemical process. The material is an active material driven by chemical fuels that drive mechanical processes in the material that lead to contraction, force generation, which induce flows. The force generation itself is regulated by chemical signals that activate force generation, and these chemical signals themselves are also transported and redistributed by flows. So this is an integrated mechanochemical process happening in the fluid.
I have one question. So here, what you show here is the one cell, the left-hand side, the figure.
Yes, it's one cell, yes.
Yeah. Then the meaning of the surface contraction means that the membrane is now contracted, or so what the meaning of the surface contraction here?
Yes, as I'll explain in a moment, there
Yeah.
is a material, a gel-like material attached to the membrane. And this
Okay.
gel-like material contracts.
I see.
The membrane doesn't really change very much its surface, but the membrane may be carried also and may also flow, but the membrane doesn't have to flow. It's essentially the material below the membrane that must flow.
some elastic material cover the cell and they contract. So that's what
Yeah,
you said.
it's
Okay.
viscoelastic.
Yeah,
It
yeah.
is and long time scales more like a fluid than an elastic material, but it's a dense, highly viscous material, which on short time scales is
So you mean
elastic.
outside the membrane, right? Outside the membrane.
Inside the membrane.
Inside. Okay. Okay.
Inside the membrane.
Inside.
It's on the
Okay.
inside of the
Now
membrane.
clear.
Yes.
Okay. Yes. Okay. Thank you.
Yes.
Yeah.
Yes. 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 And I'll come now to this subject of mechanochemical patterning as a next theme in my talk. So here's an image of this material. So this is the part of the cytoskeleton. It's made of protein filaments. And if you label them fluorescently, you can see them as these red lines. And these protein filaments interact with other proteins that are called molecular motors that can use a chemical fuel to generate forces. And they are shown here in green. Yeah, and this is becomes now a dynamic material in which these motors generate forces and drive this material in out of equilibrium state. And these filaments, they are short-lived. They turn over in about 30 seconds. So on time scales of minutes, this is a fluid, even though it's clearly a gel, and on short time scales, it's elastic. And so the picture is we have an active material, which is gel-like, but which on longer time scales has fluid properties. It's formed of filaments, which are cross-linked. And on these filaments, there can be these molecular motors can interact. Now the filaments themselves have a structural asymmetry. They have two different ends. They are asymmetric in the sense that they define a vector asymmetry. And these motors tend, when they are in presence of some chemical fuel, which is called adenosine triphosphate, short ATP, they are driven by the hydrolysis reaction of this fuel triphosphate to diphosphate and releases then a phosphate. The chemical free energy difference, Gibbs free energy difference, or the difference of the chemical potentials of these components, is the free energy available to drive the motion of motors, and they move along the filaments in the direction towards the plus end. Now, these motors can form assemblies, and these assemblies of motors can form dynamic crosslinks in this material. And now we have filaments that move relative to each other in this crosslink material. And this now introduces spontaneous stresses in the material in the presence of in the absence of external deformations or driving. So the system has what we call active stresses, and it can therefore generate spontaneous shear or spontaneous flows. And as this is a non-equilibrium system, it can also self-organize in dynamic patterns and structure. So it's an inherently dynamic material, and it's fluid-like. And let's say a little bit about the physics of such a material. The simplest picture is that we can think of a gel-like material starting from a normal passive soft solid, for example. And then if we have these motors introducing crosslinks, this will introduce force dipoles in the material. And these have to be dipoles because forces and counterforces must balance. There's no external forces, they're all purely internal forces. But since these forces can act at slightly different positions, there is a force dipole in the material. And such a force dipole, if you think of it as in the continuum picture, of coarse grain as a point object, it is a stress introduced at a particular position of the dipole. And this is a tensorial object because the dipole here has an axis. It can be characterized by a vector. P in this case would be a vector along the axis of this dipole. F would be the force exerted. A is the distance between the 2 points. So the magnitude here has units of energy. T is dimensionless. The delta function is inverse volume, so this has units of stress. And if there are now many of these dipoles in the material, in the continuum picture, this gives then a density of stress that is introduced in the material. If the dipoles are all isotropically oriented, then this is an isotropic stress, which could contract in 3 dimensions. Now, since we have incompressibility of our materials, these isotropic stresses are often less relevant and most important are anisotropic stresses, when these force dipoles are on average aligned along one axis. So if the system has anisotropies. And then the shear components of the active stress become important and can induce spontaneous shear. And now one can build sort of more generally, if we want to have a theory of such active materials, of active fluids, we can build such theories in very general ways from using irreversible thermodynamics. So starting from thermodynamics, assuming local equilibrium, and then allowing the system on larger scales to not be not at equilibrium and to exhibit dynamics. So we will start in such a general hydrodynamic approach from conservation laws, which somehow define what the slow hydrodynamic variables are that are relevant on large scales and slow times. It's energy, mass, density, and momentum. We do thermodynamics. We can also define the entropy balance, with entropies changing locally due to entropy fluxes and entropy production. So entropy has a source, it's not conserved. Now the entropy flux can be related to the heat flux, which is part of the energy flux. And if we write down entropy production, sort of based on local thermodynamics, then we can identify for different physical dissipated processes contributions to entropy production. For example, if V is the velocity gradient, that's the shear rate, sigma is the dissipative part of the stress, the product of those is the viscous dissipation. We also have a pair gradients of chemical potentials, and diffusion fluxes J, they define entropy production due to associated with diffusion. Then I have chemical reactions in my system. I just use one for simplicity, which may be the reaction that the fuel drives when it operates molecular motors. That's the chemical free energy of the reaction, and the reaction rate defines the chemical dissipation. And in general, there can be temperature gradients and heat fluxes, which also are a dissipative process. And in irreversible thermodynamics, one can now write constitutive equations for the material by writing linear relations between fluxes and forces, which are these conjugate variables. And this allows to build a very general theory based on symmetries in conservation laws. And in particular, it allows then by cross-coupling terms to couple chemical reactions to other processes, such as transport processes, such as diffusion or mechanics. And that's exactly where the activity comes in. So if I look at a very simple version of that, sort of the minimal version of that I would write a constitutive material relation for the stress, which has in a fluid, the conventional part of the velocity gradients times viscosity is the dissipative stress. And if I can couple the system via molecular motor also to chemical activity, then I can have another stress term, which is proportional now to the chemical driving force delta mu, and has this tensorial structure if the system is locally anisotropic. So P is a vector describing orientations of filaments in a coarse-grained way now, and can now describe the active stress that I introduced before in a sort of simple physical picture. And similarly, one can also couple the chemical activity of molecular motors to transport. So the currents J, which in a passive system are due to diffusion, driven by gradients of chemical potentials, can also now have contributions stemming from activity of motors driven by ATP. And this requires a vectorial asymmetry, which the filaments provide. That's why there's a vector P here. And so we can now have a hydrodynamic theory which has these active terms and captures active stresses and active transport. And this requires, as I mentioned, anisotropies in the system, which are elements of the cytoskeleton that may be anisotropically organized. So if they're bundled, then we have, for example, a system that generates such an active stress because of local polar or nomadic order in the system. And all of this is driven again by the consumption of a fuel ATP, which is hydrolyzed to ADP. This is happening via enzymes that catalyze that reaction and then couple this to other processes such as transport and force generation. Now, in order to understand the polarity breaking in the cell, we have to realize that this gel-like material is localized below the cell membrane. It's about 200 nanometer thick layer of the material attached to the cell membrane inside the cell. And so we can go to a thin material, a thin film description, and to have an effective two-dimensional material. One interesting aspect here is that the fact that we're near a surface, the system is not isotropic. So the normal vector of the surface somehow breaks the symmetry. And we can think of the polarity vector P that I introduced being biased by the normal direction. So we have, in fact, in this two-dimensional system, an example of having shear stresses in 3 dimensions, even if in the surface in 2 dimensions would be two-dimensionally isotropic. So we can now get such a effective two-dimensional description by integrating the 3-dimensional material over a thin thickness d, and then have an effective, instead of the stress in 3 dimensions, we then have a tension in 2 dimensions. And the active stress then translates into an active tension. And here we would then get such a equation for the flow velocity, which has a bulk and a shear viscosity in 2 dimensions. Gamma is a friction term that it happens, for example, in this cell because the membrane moves relative to the eggshell. So there is a rigid support, and this relative motion is associated with a friction, which I introduce here. And then whenever there are gradients of this active tension that is generated by motors, this will drive flows. And you see also that this system contains a length scale. So the viscosities divided by the friction, square root of that defines a length scale over which tension gradients have an effect. And overall, we then can write a force balance in the surface that the divergence of this tension tensor is balanced by external forces. Now, one can build a sort of a minimal physical model of such mechanochemical instabilities by combining now the physics of this active fluid with a regulation of the activity. So I'm putting now a 2-dimensional fluid on the surface of a closed surface. So let's take a sphere here. So if I put this on a curved surface, I get an extra contribution from the Gaussian curvature when I derive the 2-dimensional flow equation. So there's now a 2-dimensional fluid on a curved geometry. There are gradients of active stresses which drive flows. There may be other forces coming from the internal fluid, which I'm just mentioning here. And now I have molecules that regulate the active stress. This could be the concentration of the motors themselves, or it could be some molecule that regulates how the activity of motors. And this has a concentration C. That's just a scalar concentration that diffuses in the fluid, is convected by any flows, maybe also exchanged by some source terms from the bulk inside. And the role of the concentration C is to generate the active stress, to regulate the active stress via some function f of C, which generally has to be nonlinear in order to allow also for stabilization in case of instabilities. And T0 is sort of a strength of the activity of the system, which drives it out of equilibrium. And this simple system of coupling a stress regulator with a fluid is enough to generate patterns. So we can start with an isotropic state on a sphere, and the system now has an instability where it generates flows that localize the concentration C on this yellow patch. The yellow patch is contractile because of the regulation of contractility, and that's why the velocity flows towards this patch because of the gradients of tension, and diffusion and convection balance such that this is a steady state, non-equilibrium steady state pattern that is stable. And this happens beyond a critical driving strength below which the system, as a homogeneous system, is stable. And just a note here, you see these flows fields in 2 dimensions have a divergence, even though the material in 3 dimension may be incompressible. So the 2-dimensional divergence being not zero implies there are then flows that are perpendicular to the surface and go inside the sphere. So the 2-dimensional material is compressible, while the 3-dimensional material may not be. And usually we use an incompressibility approximation in 3 dimensions. Now, this motion on the surface can now, by no slip boundary conditions, be coupled to a fluid inside, as in the cell interior is fluid, but mostly passive in its effective behaviors. And then we get this surface flow field as shown here with a high concentration on one side, and we get these slow patterns inside. And this is the steady state of this very simple model, but it captures some of the basic features of what happens in the cell symmetry breaking. It's a polarization of the surface, and it generates the flows that are observed inside the cell during asymmetric cell division. But of course, it's a primitive physical model system. It doesn't really capture any details of the cell. If one wants to describe what happens in the cell, one has to add some complexity to the problem, and I just want to show some ideas here that are relevant. So we have now quite a good quantitative understanding of the symmetry breaking of the cell I showed you before. One can essentially capture it by an active fluid, where flows are driven by gradients of active stress on the surface. And I show here, for simplicity, a 1-dimensional model, which is of this problem. But here we add more components. So instead of having just one variable that diffuses on the surface, we take the 2 different proteins into account that are here labeled in red and green, called par 6 and par 2. That's why they have these indices. Each of those diffuse, and described by diffusion coefficients. They are convected in the generated flow, but then they can also be exchanged with the cytoplasm inside. They are on and off rates. And what stabilizes this interface between red and green are coupling interaction terms, where one molecular species drives the off rate of the other molecular species, and therefore they exclude each other on the membrane. In addition, we also capture the concentration of motor molecules, because we can also measure and observe this directly on the surface, which also diffuse, which are also convected. They can also stochastically bind and detach from the surface and be exchanged with the bulk. And the active stress is then a function of the motor density. So that's a more complex model, which is somehow meant to capture some of the details that we can observe in the experiments. But the underlying principle is sort of more captured by the minimal model I showed you before. And just to say a few more words about how symmetry breaking happens in a cell. This system is now triggered and guided by cues, and the cues come from the entry of the sperm, which has to pass through the cell membrane to get inside, and therefore perturbs locally the membrane and perturbs some of these equations with some position and time-dependent pulses. And these position and time-dependent cues from the sperm entry now drive the switch from a symmetric state to a broken symmetry state in the cell. And this is illustrated in this examples. These are sort of 1-dimensional solutions to this type of model. So the system is a switch. It has actually 2 stable steady states. It can have a symmetric steady state, which is shown here with the surface has now this red component homogeneous everywhere, and the green component is 0 because it's in the inside of the cell. And on the left side, the black line, there are no flows. Now there is the sperm entry now triggers a perturbation, and that leads then to gradients of tension, active tension on the surface, which drive flows, which you see on the left. And this now pushes the domain to the side and allows the second domain to form. And then the contractility is switched off, and the system relaxes to the second stable steady state, which is polar. And this state with these two caps is a steady state that is stable and doesn't need flows to be maintained. The flows are there to switch from the symmetric to the polar state. So we think of this mechanochemical system as a sort of containing a self-organized pattern formation system, which can either be symmetric or polar, and then guiding cues that drive the toggle the switch realized by the system. So we call this guided pattern formation. So we can think of a system as a dynamical system that is self-organized and forms patterns that has different steady states, which are fixed points in a dynamic systems perspective. One of these fixed points corresponds to the symmetric state, one of these fixed points to the polar state. These fixed points have basins of attraction, which are separated by some line. And now the guiding cues which trigger these flows, they can drive the system away from its fixed point by some genetic program. And if it doesn't drive it far enough, it would go back to the initial state. But if it drives it far enough that it passes this green line, the system would be in a different basin of attraction and would then find the other state. And that's driven by the guided cues, in this case by the sperm entry, to reliably toggle the switch in a particular direction. And one can play with the system experimentally by perturbing, for example, the strength of the motors. And this can be done by sophisticated biochemical and genetic techniques. And here, this then leads to a change in the flow speed, which is measured here in different cells. So each dot is a cell. And these are non-perturbed ones, and these are perturbed ones. And one can then see that, let me see, these are the perturbed ones, these are the non-perturbed ones. And one can see that if the flow speed goes beyond a certain critical value, then the polarization does no longer work. And the system sort of stays within the symmetric fixed point, even though there is some attempts to move it a bit away. Okay, so that was a bit biology. Now let's go back to the physics. I've shown you that we can sort of boil it down to a much simpler model, a minimal model for this polarity establishment. And we can draw a stability diagram for this model. So there are two important parameters. One parameter is how far the system is driven out of equilibrium. This can be characterized by a Peclet number. Peclet number measures the relative importance of convection and diffusion. And this Peclet number is essentially proportional to the active stress that is driven by motors. The second variable is the length scale that I introduced before, relative to the size of the sphere. So this length scale can be small or large compared to the sphere diameter. It can be either the ratio of the viscosity and the friction, and some version of the model is the ratio between the three-dimensional and the two-dimensional viscosities, depending on which one dominates. Now I showed you that there is an instability towards this polarized state, where we have a patch of concentration on a tip and flows towards this patch. And in the calculation where one looks at the stability, one can use spherical harmonics to describe symmetry breaking. And then this symmetry break corresponds to the instability of an L equals 1 mode in terms of spherical harmonics. Now one finds in this analysis also that the system can get unstable with respect to an L equals 2 mode. If the hydrodynamic length is shorter and the system has to be driven a bit more then. And in this regime, where the L equals 2 mode is unstable, instead of a vectorial asymmetry, it generates a nematic or tensorial asymmetry, which has an axis, but not a vectorial direction. And this corresponds to the formation of a ring. So I showed you this example here. We start again from an isotropic state. And now the system instability leads to a formation of a band of concentration around the equator and flows moving from both sides into this band. This is a nematic or polar instability. And that's, one can think of as a simple model system for the formation of a contractile ring needed to divide a cell, because you can imagine that if you have a ring of contractility, if you allow the shape to change, the mechanics will lead to an indentation. So one can and now use this instability to change the shape of the cell and to start invaginate to use division. So this here is just shows a linear calculation of this process. More recently, colleagues here in Dresden have numerically solved this problem to really generate a division of such a spherical object using this stability. So this simple model system can account for polarity establishment, but can also account for ring formation, allowing for division as a simple toy model in physics. Okay, now let's come to the next aspect that I can again discuss with asymmetric cell division, still staying with my simple model example. And that is the fact that in addition to the cell surface becoming polarized, also the cell interior becomes polarized in the process. And there are first the different surface properties are used by the cell to generate concentration gradients inside a cell. And that's one protein highlight here is called MAX5, which is shown here in green, which has high concentration on the interior side, low concentration on the posterior side. And it can be shown that this protein gradient organizes the segregation of these green objects, so-called germ granules, that are segregated to one side of the cell, and as the cell divides, one daughter cell gets these germ granules and not the other one. And this brings me to the problem or the issue of active emulsions in the cell, as these germ granules are droplets, and the segregation is an example of emulsion physics.
Professor, I have a question.
Yes.
What happens if two sperms enter the cell simultaneously? Then in that case, this process, how this process could be affected, and then this model, how model can be changed.
Yeah, I don't know if this can be tested experimentally, because the cell would only allow one to enter. So I don't know if this can be done experimentally. That's an interesting question. But it probably requires some tweaking, but I'm not sure if it's possible.
Okay.
Because usually the moment one sperm enters, the cell quickly is protected against the entrance of a second one.
Right, but the fraternal twin case, the two enter actually. It's very rare case, but I just curious what will happen in the full dynamics.
Okay, I don't know.
Okay.
Yeah. So okay, so here you see the same cell as before, or let's say same cell type, the first division of this fertilized egg, but now a different protein labeled. The protein called PGL1. You see it in green, and this clusters in these small assemblies called P granules. And these assemblies are condensates containing RNA and proteins, more proteins than PGL1, but there's a lot of PGL1 in them, as you can see, as there are these spots. They come in different sizes. And initially, when the cell is not polarized, so the membrane outside is now always everywhere the same, they are more or less equally distributed in the cell. And as the cell polarizes in this process I described before, they then now start to segregate towards the right. Let me show that. And now they're segregated, now the cell can divide. Now, it looks by eye as if they are segregated by the flows that I introduced before. And this is actually deceptive. If one quantifies this process, one can see that the transport by flows goes in both directions, because the flows are circular, as I showed you before. And there's the fluid flow, because of incompressibility, is there a flow to the right, there must be counterflows to the left. And this sort of equals out roughly. And there's almost no contribution from flows. The segregation happens via growth and disassembly of these objects. Let me say a few words about these P granules. These P granules are segregated during asymmetric division, always to the cells which are the precursor of the germ line. So in the first division, they go from P0, they go to the P1 cell. In the next division, asymmetric division, they go to the P2 cell. In the next asymmetric division, they go to the P3 cell. And then these are the precursors of the germline. And in the end, they are only found in the sperm and egg cells of the in the gonad of the adult animal. These are assemblies of proteins RNA that there's no membrane around them. These are so-called membraneless compartments. They are in the cytoplasm of all the germ cell precursors, and they are implicated in what makes germ cells, yeah, which are different from somatic cells that die with the individual. This germ cells somehow immortal and live on from generation to generation, and they're specified by these RNA protein structures called P granules in the worm. Now these P granules, when one observes them carefully into the microscope and realizes them, but Cliff Brangwyn, which realized that when he was a postdoc in Dresden with Tony Heimer and myself, they behave as liquid droplets. So one can see, for example, at a microscope, P granules, when you can touch and fuse, and they fuse then to become spherical, which hints at some surface tension. Here on the right side, you see an adult worm. The circles here, the big circles, are nuclei in the gonad of the worm. And these nuclei on the surface have material of these P granules, which are these labeled PGL1 proteins. And so they form droplets on the surface of the nucleus and wet the surface of the nucleus. And if one now squishes somehow the material, the worm, then there are flows generated and they deform as liquid droplets would. So from such experiments, one can estimate the viscosity and the surface tension. One finds that the viscosity is quite high. It's, let's say, 1,000 times the viscosity of water. From the relaxation times of fusion together with diffusion coefficients, one can estimate the surface tension. So one can characterize them as physical droplets in the living cell. One can purify components. This is another component of this P granules, purified in buffer solution, and in certain concentration regimes, they form just in systems, just one protein in buffer, they form emulsion droplets. And you see such an artificial emulsion made of these PGL proteins in the test tube. And you see that emulsion ripens, droplets can fuse. And so this is classical emulsion physics that we see here. So proteins can assemble in dense liquid phases that coexist with the outside. And in context of the cell, this often happens in a multi-component way, many proteins together, typically also with RNA, forming such liquid phases inside the cell cytoplasm. And these structures, such condensates in the cell, where proteins and RNA form small droplets, key examples what are often called membraneless compartments. So biochemistry in the cell is organized in different chemically distinct regions or compartments, which classically are separated by membranes, such as in the typical organelles that have different functions inside the cell. But there are also many of such compartments or organelles which have biochemical function in the cell that are localized objects that do not have a membrane, and they are often liquid-like and often can be thought of as phases that coexist with the surroundings. And here's a number of examples of that. The upper left, we see cell nuclei in red, and in green we see compartments called nucleoli in green that are also actually liquid droplets inside the cell nucleus. Other types also in the cell nucleus are these Cajal bodies in green. Here are other different types of objects, and these are all identified by labeling a particular type of molecule, which are then localized in such small structures, and that's a way to see the spatial organization in the cell, that it has many of these granule-like or droplet-like structures which condense certain types of proteins together in a small structure, which can serve as a chemical environment where certain processes take place that elsewhere would not take place. And from the point of view of physics, one can think of this as the coexistence of different liquid phases that phase separate. So the simple physical picture is that we can have a complex mixture in a liquid, and if this undergoes a demixing phase transition, then we would have phases of different composition that coexist at identical chemical potentials, and we can draw a phase diagram as a function of composition and temperature, which has a demixed region and different mixed phases. And of course, if we then now carry this over to a biological problem, it's important to understand that this phase separation can be regulated chemically, for example, by composition, but it will also be temperature dependent. And of course, we have also the physics of emulsion. So liquid droplets may grow or shrink and exchange material. And all of this happens in a non-equilibrium context in the cell, as I will also talk about in a moment. Now, in the case of the P granules that are segregated, the segregation is driven by a protein concentration gradient that somehow regulates assembly and disassembly of these objects. So on the anterior side, where there's a high concentration of max5, they will dissolve. On the posterior side, they will assemble. Here you see a two-color labeling, where we see max5 and P granules at the same time. So the max5 has higher concentration on the left and on the right in the course of this movie, and the P granules segregate from the left to the right by dissolving on the left and growing on the right. And in the sort of physical phase separation picture, and this is very schematic here. This is not meant to be a detailed model of the process, because it's difficult to sort of plot a appropriate phase diagram. So this would be a phase diagram which has the P granule material density on the horizontal axis and a regulator, the max5 density, which is the concentration gradient on the y-axis. So for high max5 concentration, we would be out of the phase coexistence region. The green line is a binodal. The straight tilted lines are so-called tie lines, which connect coexisting phases. And if we look at the non-polarized cell, we would have a certain average concentration of material. If it's in the coexistence in the phase separated region, the dilute and the dense phase are the ends of this tie line, which corresponds to the droplet, which corresponds to the dilute phase. If now the cell breaks its symmetry, we have now a gradient of max5, so the anterior and posterior side have different max5 concentrations. That's why they select different tie lines. So the dilute and droplet phases are now selected by different tie lines. The dilute phases have different concentrations, and therefore there is a concentration gradient in the dilute phase across the cell, which drives the diffusion current and transports material from posterior to anterior. And I see that we're already at 1 hour. I just want to ask how much time I can still take, whether we are still flexible.
So I think we have a 15 minutes.
15 minutes, yeah?
Yeah. It's okay. Yeah.
So it's okay if I continue a bit more, yeah? Okay.
Yeah. Yes.
Good. So now we can ask the question whether such a picture of phase diagram and phase separation is appropriate. And the first test to do that is to ask whether temperature is a thermodynamic control parameter of this process. So one can change temperature in the system. So in the top, I show you a cell where the temperature is increased, and below it's a control where the temperature is unchanged. And increasing the temperature actually dissolves these P granules. And when this is reversible, one can lower the temperature, and they reappear. And in this case, they then also reappear on the appropriate side, because now the max5 gradient has formed, and they actually grow only on the right-hand side here. And now this is also can be repeated. So here you see temperature changes, temperature going up, going down, going up, going down over time in the experiment, and the P granules dissolve and form and dissolve and form repeatedly. One can now measure concentrations inside and outside of the droplets using fluorescence intensities at different temperatures, and draw something which looks like a phase diagram. So these are the concentrations of the dilute phase. These are the concentrations of the dense phase, which depend on temperature. And then we can also sort of capture this behavior by a very simple, effective binary model of phase separation. Now, this raises, of course, the question whether it makes sense to use an equilibrium phase separation picture inside a living cell, which is far from equilibrium environment. And I just want to give you some order of magnitude ideas about this question. First question one can ask is, can one define properly a temperature in a cell? So clearly, if I think of temperature, I can write down a diffusion equation for the heat transport. And there will also be source terms because the cell releases heat by its biochemical activity. One can show that the temperature relaxes very quickly in the cell of this size, given the diffusivity. So it's easy to impose a temperature from the outside, and the heat source is weak enough that it doesn't perturb really the temperature profiles. What is sort of more subtle and more interesting is to think about the fluctuations, because the heat release comes by stochastic individual chemical processes in the cell. And one can think of an ATP hydrolysis event as a stochastic, very local event, which releases about 10 kBt of energy in form of heat, which is locally a very strong perturbation as a non-equilibrium effect, but this dissipates in a very short time scale, taking this order of magnitude, let's say, of thermal diffusion in water. So about a microsecond after the event, this has already spread out over about 100 nanometer, and there's no temperature effect left. So if you look at timescales longer than microseconds, all slower processes will not really see this effect, and the system is sort of a length scale of 100 nanometer, and timescales beyond microseconds is in the local equilibrium to some good approximation. In particular, if you, for example, look at an interface of a condensate, one can think of a local equilibrium as a good approximation, and then the non-equilibrium effects somehow happen at longer time scales and length scales, because different locally equilibrated volume elements are not at equilibrium with respect to each other, and this gives then rise to this irreversible thermodynamics, as I've introduced earlier in the talk. So this is an example of a stochastic simulation of an emulsion segregating in a saturation gradient to mimic what happened in the cell. And let me see if I have a few more minutes. Maybe I'll still talk a bit about chirality. I introduced that in my introduction, that cells can be polar, but they can also be chiral. And one can actually observe directly, again, at the single cell level, one can observe chiral asymmetries in the flows. So if we observe now in this case, we see the motor density, and we can observe these flows in average over many cells. And here you see the horizontal velocity profile due to polarization of the cell. On top of this horizontal velocity profile in the x direction, there are also flows in y direction, and these flows are actually chirally asymmetric. This is not visible by eye in this example, but there are particular mutations and perturbations where this is highlighted very strongly, as I show you in this example here. Here you see these chiral rotations pronounced, and this is a mutant of a single protein. But the same chirality is also present in the normal case, it's just not very pronounced. And the origin of chirality is the fact that filaments are not just vectorial asymmetric and then allowed to introduce false dipoles. They also have a helical structure, and the molecular chirality leads to this helix of filaments. The helix of filaments introduces then also torques and torque dipoles in the problem. And I can also introduce such chiral active processes in my active fluid model by having an extra term due to these torque dipoles. And this then gives rise, if I look at the symmetry breaking, I still have my polarization, my patch on the surface, but I now get chirally asymmetric flows which have this vorticity towards the pole. And the sense of chirality is set by the chiral asymmetry of this extra term in my equation. And this can be used to discuss the chiral symmetry breaking of the embryo. This is not due to cell chirality in the first cell stage, because this division can only be used to break the one mirror plane. But when we go to the 4 to 6 cell stage, the cell division of these cells along the left-like axis, which then changes the angle, this can be accounted for by chiral flows on the surface. So here you see actually this process happening. These cells divide, and then from the axis was initially in the LR left-right direction, tilts, and then the embryo is left-right asymmetric. One can measure now the chirality of the surface flows of these cells and reveal this chirality. In a model picture, this would correspond to the symmetric case, which forms a ring, but with extra chirality, it generates the counter rotations on the two halves of the cell. And if the two cells sit in an asymmetric environment in the embryo, then they can lead to rotation. You can think of the two ends of the cell to counter rotating. This happens in an environment which has already broken symmetries from the previous cell divisions, like two spheres rotating on a flat surface, and then the whole object will have a rotating axis, similar to an excavator. If it counter rotates, its chains can rotate on the spot. And so this accounts for the left-right symmetry breaking in the worm, which is governed by surface flows and chirality of the ectomyosin of the cytoskeletal system on the cell surface. Just a short comment about how this happens in mammals, including humans. There are also left-right symmetries broken, but this works by a different system, but also by the cytoskeleton. Instead of using the surface flows of the cell, in the case of the worm, in mammals, it is beating cilia. These are whip-like structures which can move, driven by molecular motors, and which can generate rotatory motion, and then generate left-right asymmetric flows of a fluid. So in all these cases, we have active processes in the cytoskeleton that are chirally asymmetric, that drive flows that break left-right symmetries of organisms, but slightly different processes in worms and in mammals. And my time is essentially up, but let me just a few words for the last part, which I keep short, bring me back somehow to the problem of droplets and condensates in cells. We have used that to think about droplets as a very simple and very old aspect of physical chemistry that was probably relevant also very early in evolution, and maybe can be a guide about what early cells looked like when life emerged at the origins of life. Because, of course, we don't know much about that, but you can think about what is the simplest physical chemical systems that can generate lifelike features. And looking at droplets driven by chemistry is an appealing example. So that I'm taking a phase-separated system. I also introduce chemical processes which turn over the components that phase separate in a reaction diffusion type of system. The system is driven out of equilibrium by some fuel. It could also be driven by light or by other energy sources. And this gives rise to emulsions with unusual properties. So instead of just ripening, they may undergo arrested ripening and have droplets of well-defined size that are non-equilibrium steady states and behave very different from passive droplets. And in addition to just being non-equilibrium steady states, they can also undergo shape instabilities and actually divide. So this is the minimal model, physical model of division of droplets. And and we think of these systems as minimal models of cell-like structures that are out of equilibrium, that are turning over like in a metabolism of a living system. They have well-defined shape and size, and they can undergo rounds of division and growth, mimicking some features of living cells. They are sort of a minimal model for a protocell. They can undergo growth and division. They are far from equilibrium, have to be maintained there in order to have these behaviors. But of course, they do not contain any genetic information and therefore do not undergo evolution, but it's something one could think of adding as complexity to such models. And with that, I think I'm very long in the time here, so I should probably.
Okay, thank you, Professor Jülicher. So it was very inspiring. And in the beginning, I was very curious, and then I learned that you explain one by one about the introduction, what you explained to us. So so it's a question time. So yes, please, if you have any questions, so you can give a question, or you can also using the chat mode, so you can maybe typing your question, then yeah, I can read, or he just read it and then answer. Yeah.
Can I start?
Oh yeah. Yes,
Okay.
please. Yes.
The Professor Frank Jülicher, I really enjoy your talk. I thank you for your wonderful and insightful talk. And actually, I at first thought there should be no reason to break left and right symmetry, because forward and backward and up down, I think I can understand that there should be ample reason to break the symmetry. And then I don't see why left and right has to be broken. And then I was wondering what would be evolutionary.
Hello, can you hear me? I'm sorry, I think I've got disconnected. Somehow I lost the connection.
Okay.
Okay. So, yeah.
Right. So
I didn't realize. I don't know when the connection broke down. What was the last part that you saw?
Okay.
다시 한번 얘기해주면 될 것 같습니다.
Right. Okay. So I really enjoy your talk. I thank you for your wonderful and insightful presentation. And then at first thought, I think there is no reason for breaking left and right symmetry. I can understand that the forward and backward and up down symmetry can be broken for many reasons. And then is there any evolutionary advantage of breaking left and right symmetry, or just this is a mere consequence of some spontaneous breaking at some moment? What is your
Yes, so we know that symmetry is broken precisely in some sense. For example, the heart in a mammal is always on the left side, and there is, I think, is only 1 in 10,000 individuals had it on the wrong side, and usually that's associated also with other issues. So the symmetry breaking is somehow controlled biologically, but there are also different processes that break the left-right symmetry separately. For example, the internal organs break the left-right symmetry independent from, for example, the brain. So there are at least 2 different processes which break left-right symmetry and that are independent. Also, I've shown that in different organisms, slightly different ways are used to do it. So it seems to be a common feature that biology does break it systematically. Now, on the surface, let's say the outside of the organism, you may not want to break it because it hinders locomotion. Yeah. So if you want to be able to move around, it's difficult if you have too many broken symmetries, because you could not move straight. On the other hand, to build a complex organism, it may be useful to have the degree of freedoms of having more flexibility of the but I can only speculate, so I don't have a good answer. One idea is that if you think about the brain, if it's perfectly symmetric, you would have a hard time to respond quickly in some situations where you then don't know which way to break your symmetry, when you have to escape left or right. So if you have broken left-right symmetry in the brain, it may help you to be fast.
I see. Can I have one more question? Okay, so you also mentioned the chirality, and then the
Yes.
part actually rotate this way, the other part rotate the other way, right? And then is it this chirality is actually can make it can be useful to for in cell division? Okay, it's breaking rotating this way so that cell division is can be more facilitated. I just very simple idea. Maybe the chirality
Yes.
is actually something to do with the cell division. What is your
Yes, that's an interesting idea. I haven't thought about it. So so we see chirality in the very first cell division even, but there's nothing left of it later. It could be helpful. I don't have a concrete idea about it, but it's a fascinating idea. Yeah.
Thank you.
Okay, so any other questions from the audience? You may ask some very general question to him if you want. Yeah.
Oh, I have one question.
Yes. Yeah.
Oh, thank you for the great talk. Uh, in the first part of the talk, there was like this regime where the flow was towards the center, so the contractile ring forms, and there was also regime where the flow was towards the left, so the asymmetric distribution of these membrane proteins. But I think those two events or those two regimes should occur at the same time during the cell division. Is that right? So, have you
Yeah, of
seen?
course, the model with these two cases is, of course, oversimplified when it comes to the biology. So the polar version is is close to what happens to polarization, and the symmetric one may be close to what happens in symmetric cell division. Now, how to combine the two, we don't have a good model for it yet. It has to be more complex. Yeah? But when you want to have the polarity of the domains and the ring at the interface. Yeah? Now, in the cell, it's also that the positioning of the ring is also influenced by the spindle. And of course, my simple model doesn't include that. So in order to build a model of the cell that includes the formation of the ring, one probably also has to add the spindle to it. And the spindle then helps to bring a ring in the center, even if the symmetry is broken.
Okay,
And
thank you.
there's a number of interesting questions about how to do that, because if the symmetry is broken, there's a bigger danger that the ring slips to one side when you try to divide, because it's asymmetric. It's harder to maintain it in the center, so you need to re-stabilize it very much. And the spindle could play an important role there.
Right.
I have one very general question in the beginning. So you explained that how the embryo develop. Then it's symmetry breaking is very important, and you show that one cell divided by two, and one cell is preserving the symmetry, the other one breaking the symmetry, and then how this development is robust. So I thought that it is very so if you're doing this experiment repeatedly, then they're always the same coordinate like that.
And this depends very much on the system, yeah. So what I just also I focus on one particular biological system,
Yeah,
also to keep the discussion somehow
Mhm.
simple, and also
Mhm.
because we know a lot about it. But in this particular system, this is really robust, and it happens that way. So the very first cell division exactly breaks the anterior posterior similar. The next division breaks exactly the dorsoventral, and the next one break the left right. In mammals, this is different, yeah, and also in insects, things are very different. And in mammals, you first have a clump of cells that are very similar, and then you break the symmetry a bit later when you have already a collection of cells. In flies and insects, you first, when you form the embryo, you also create many cells first, and then you start revealing symmetry breaking and so on. So in biology, you often have to look at different systems, and you have quite different scenarios. But from the physics side, there's still The principles are still the same, that you have this active matter that uses cytoskeleton, but the way these programs that run in detail are different. And in the worm, it's a special case where even the number of cells in adults are always the same. The number of neurons are the same, and all these. That's also why all the cells have names. So there are a certain number of cells, and they all have names. One knows exactly where they appear. This is different in other species where you would not have this precision.
Then, when this order of symmetry breaking is not what planned, then it means that something, some fatal thing happens. So the embryo development is going some strangely. So that's what we expect. Probably.
Yeah. So if this doesn't, if this is perturbed and it doesn't happen, then this doesn't develop to an adult. Yes.
I see. But this kind of then, which means that the cell-cell interaction could be also another important interaction to making this order of symmetry breaking. That it is some kind of a new problem that we don't understand right now. Yeah, I see.
Yes.
Okay.
Because the whole thing has to be coordinated. You're right. Yes. So if you look, that's why it's nice to look at the first cell division. That we don't have to worry about the others.
Yeah.
If you go to more, then they interact, and then they have to coordinate. So and I don't think it's understood how this works. Yeah, that one cell only divides after the neighbors have done the right thing. That because there must be spatial temporal coordination, and that seems to be achieved in this worm. And I don't think we understand how this works.
I see. And the other some general question is, the cytoplasm is a mixture of a polymer and some many molecules, so that in general it's like a viscoelastic fluid. So then I think this effect is how much important, because it seems to me that this effect is not encoded in your theory, right now. So how much this kind of viscoelastic effect could affect this, the symmetry breaking?
Yeah, that's a very good question. We don't know too much about the rheology inside the cell. Of course, one can make cell extracts and measure properties there. So my understanding is that if you go on time scale of the minutes, then you have a fluid, in the sense that you can define the viscosity, which is quite large. On shorter time scales, you see the elasticity. There could be longer term elastic effects as well, but I don't think this is well established. And so there is a lot of potential for interesting effects in physics there, but we don't know enough about them to include them in our models.
I see.
And so our models are also built in a way that we are being as simple as possible. And we try to be robust in the sense that if we're missing things, we still capture them generically, but it would be nice to know more about this.
Yeah. I fully agree that. Yes. As a theorist, we should start from the minimal model. We understand that level. I fully understand this approach. So I think the time is now quite what we expect, almost 90 minutes. So is there any questions from the audience? Then I want to have one more question if you have.
I have a question.
Yeah. Okay.
Yeah,
Yes.
thanks for the nice talk. And I have a question regarding the growth. I mean, you assume that your system is incompressible, which, as far as I understand, this means the cells are not growing, but I guess cells are growing. And the polarity is also broken in the process of cell growth. I mean, how this division and cell growth can be like incorporated into like single framework? Is there any idea
so
or
it's an interesting point. So first, I'd like to note that, of course, cell growth is a very interesting phenomenon, but it doesn't imply that it's not compressible, because the material is probably close to incompressible, but you have to add material. So you have to transport material inside the cell to grow the cell. So cell growth is always associated with transport of matter into the cell. Now, in the case of the egg, everything is inside. So there are gases exchanged. So oxygen goes in and CO2 goes out, but essentially everything that you need to build the organism is inside the egg. So in that sense, there is no growth. And as you develop the embryo, you just subdivide volume. So the first cell is big. That's why it's also a good cell to work on as a physicist. Then it divides the two cells. They are each half the volume, and then with more and more divisions, the cells get smaller and smaller. So there is no cell growth, and then only after this hatches it can grow. And it grows by taking up material from the outside. This, of course, again different. Let's say in mammals, where you can at the beginning also you subdivide cells, but later you grow them. But it's an interesting point, and we do include growth in our theories, but it has to take into account material conservation. So you have to bring in material.
Thank you.
Okay. Do you have further questions, Segon? Otherwise,
yeah.
yeah.
Okay. Yeah.
Okay. Otherwise, I think time's up. So I really thanks again to Professor Jülicher for very nice talk. And also thanks to all the audience of this colloquium. So, okay. So bye-bye. And I hope to see you again, maybe next year, right? Yeah.
Yeah, thank you for the kind invitation again. It was wonderful to see you.
Yeah. See
And I'd
you.
love to visit at some point. Yeah.
Oh,
Bye-bye.
yeah, sure. I'll visit you. I invite. Yeah, okay. Okay, have a nice day. Yeah, in Germany.
Yeah,
Okay.
bye-bye.
Yeah, bye-bye.
Bye-bye.
Yeah.